Welcome back to the deep dive. Today we're doing something a little different.
A little bit.
We're sort of putting on our white coats, step into the role of medical educators.
We are, because we are tackling the fundamental machinery of life itself.
That's it. We're diving into Essential Biochemistry, 4th edition. But before you tune out,
Right.
thinking this is just, memorizing chemical structures or stressing over the Krebs cycle.
Let me stop you there. This is the discipline that answers the two most important questions in medicine. What are we made of? And, maybe more importantly, how do we work?
Exactly. Our mission today is to take this huge textbook and just synthesize it, turn it into a survival guide for a medical student, a young professional, or just anyone who's intensely curious.
And we're not just listing facts. We're looking for those aha moments, the things that explain clinical realities.
Yeah, what's the why behind it all?
Precisely. We're going to look at the chemical foundations, the dynamic structures like bone and blood, the engine of metabolism, and the genetic code, but always through the lens of why it matters when you're treating a patient.
So, okay, let's set the stage. We're talking about the molecular logic of living organisms.
And what's wild is that whether it's a single-celled amoeba or a human being, the chemistry is surprisingly universal.
It all starts with the periodic table, right? Which in a chemistry lab is this huge, intimidating thing.
It is. It's vast, over 100 elements. But the periodic table of life is incredibly exclusive. Biological molecules are primarily built from just 6 elements.
The big 6. H, C, N, O, P, and S.
Hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. That's pretty much it.
It's amazing to think that almost everything in a cell, your DNA, your muscles, your brain, is just a different arrangement of those few ingredients.
It's an elegant simplicity, really. And from those 6 elements, we get the 4 major classes of biological molecules. If you're going into medicine, you have to know these 4 players intimately.
Okay, let's break them down then. Who's in the lineup?
So, first up, you have amino acids.
The building blocks of proteins.
Exactly. The workers, the structures, the enzymes. Second, you have nucleotides.
And those make up DNA and RNA, the information?
Your information storage, yes. Third, you've got monosaccharides.
Your carbs and sugars for energy and structure.
Right. And finally, lipids.
The fats.
The fats. Crucial for membranes, the borders of the cell, and for signaling. Now, here's a key distinction that trips people up.
Okay.
3 of these groups form polymers, long chains of repeating units. Amino acids link up to form proteins, nucleotides form DNA, monosaccharides form polysaccharides.
But lipids are the odd one out?
Exactly. Lipids don't form polymers in that same way. They aggregate to form membranes because they're hydrophobic, they hate water, but they aren't those long, covalently bonded chains you see in the others.
That's a great way to put it. The book uses a really good analogy for why we study these things together. Something about a clock.
Oh, yeah, I love that. If you take a clock and you just disassemble it, spread all the gears and springs on a table, it no longer looks like a clock.
And it certainly can't tell time.
It can't. So studying molecules in isolation isn't enough. Biochemistry is really about how all those parts cooperate to make a functional unit.
We're looking at the clock ticking. And speaking of functional units, let's talk about protein structure. The mantra is form follows function, right?
Always. A molecule's physical shape is everything. If you change the shape, you change what it does. And proteins are the absolute champions of this. They're the most structurally variable and functionally versatile molecules we have.
And the classic case study for this is always hemoglobin and myoglobin.
It's the perfect example. And you have to give a nod to John Kendrew, who did the painstaking work to figure out the 3D structure of myoglobin. And funnily enough, when he finished, some people were disappointed.
Disappointed? Why?
Well, they expected this beautiful, simple symmetry, like the DNA double helix. But myoglobin was just irregular, complex. It looked messy, almost like a wadded-up piece of gum.
But that messiness was the point.
That messiness was the highly specific engineering that creates a perfect little pocket for oxygen. And that's also where things can go wrong.
Let's get into that, the clinical connection here. Carbon monoxide poisoning. Why is CO so much more dangerous than other gases?
It all comes down to affinity. Carbon monoxide binds to that pocket in hemoglobin with a much, much higher affinity than oxygen does. It basically bullies oxygen out of its seat on the bus.
So even if there's plenty of oxygen in the air, the hemoglobin just ignores it and grabs the CO instead.
It does. And the source makes a great point that this isn't just about big things like house fires. It's about incomplete combustion.
From gas appliances, cars.
And smoking. This stat was
Yeah.
sobering. In heavy smokers, the level of carboxyhemoglobin, that's hemoglobin bound to cocaine, reach up to 15%.
So 15% of their oxygen carrying capacity is just gone, offline.
Exactly. They are functionally anemic. It's a silent threat because it shows how a tiny change at the molecular level can have massive systemic effects.
And speaking of tiny changes, what about hemoglobin variants? We hear about sickle cell, which is devastating.
And sickle cell is caused by a single amino acid swap, which is incredible. But what most people don't know is that about 7% of the world population carries some variant gene for hemoglobin.
7%, that's a huge number.
It's massive, hundreds of millions of people. For biochemists, these are like natural experiments. Most are benign, but by studying how a tiny change alters the function, we learn how the protein is supposed to work.
That's fascinating. Okay, I have to bring up my favorite fact from the reading. Crocodiles.
Ah, yes, the crocodile hemoglobin.
Humans can hold their breath for a minute, maybe two, if you're lucky. A crocodile can stay submerged for an hour, and this is a biochemistry trick.
It is. It's an adaptation we just don't have. They use bicarbonate HCO3- is something called an allosteric effector.
Okay, unpack that. A allosteric effector.
It just means it binds to the protein, but not at the main active site. It binds somewhere else. But that binding action changes the protein's overall shape.
And for the crocodile, that means...
It means the bicarbonate binds to their hemoglobin and forces it to dump its oxygen into the tissues much more efficiently than ours can.
So they can squeeze every last drop of oxygen out of their blood while they're wading underwater.
That's exactly it. It's molecular engineering, custom fit for their ecological niche.
Incredible. Okay, let's move from the swamp to the skeleton. The dynamic body.
This is such a critical concept.
Yeah.
We tend to think of bone as this static, rock-like scaffolding,
Right.
but it's not.
Yeah.
Bone is constantly being torn down and rebuilt. It's incredibly dynamic.
This is the battle between osteoclasts and osteoblasts.
Exactly. It's all about balance. The osteoclasts are the demolition crew. They release enzymes and acid that digest the old bone. Then you have the osteoblasts, the construction crew, that come in and build new bone.
And the coolest application of this is orthodontics. Braces.
Yes. Have you ever wondered how braces actually work? You're not just bending the teeth.
You're applying stress to the bone.
Precisely. That stress triggers the remodeling. The body senses the pressure, recruits osteoclasts to dissolve bone on the side being pushed,
Creating space.
and then osteoblasts build new bone on the other side to fill in the gap. The tooth is literally dissolving its way through your jaw.
So braces are just a biochemistry hack.
100%.
Yeah.
But there is a dark side to this dynamism.
Cancer.
Yes. The book notes that these active remodeling sites, because they have so much blood flow and turnover, can unfortunately be access points for metastatic cancer cells. Breast cancer, for example, often moves to the bone.
Wow. So the body's own repair system can be hijacked.
It can. Now let's shift from solid tissue to liquid tissue, blood coagulation.
One of the most complex cascades in the body.
It has to be. You need it to clot instantly when you're cut, but you can never have it clot when you aren't. It's a razor's edge.
And the solution is something called a zymogen.
A zymogen is just an inactive precursor. Think of it like having the safety on for a whole bunch of enzymes. They're just floating around inert.
Until there's an injury.
Right. An injury triggers one to activate the next, which activates the next, and a domino effect that ends with the enzyme thrombin converting fibrinogen into fibrin.
And fibrin is the actual clot?
Fibrin is the mesh. It's the net that stops the bleeding.
So the clinical connection here has to be warfarin. It's a super common blood thinner.
Yeah.
How does it stop this cascade?
Warfarin works by inhibiting the enzymes that recycle vitamin K. Without vitamin K, the liver can't make the final modifications to the clotting factors. So essentially, it prevents the safety from being taken off the gun. The factors are there, but they can't be activated.
But dosing warfarin is really tricky, right? Different people react very differently.
And that gets us to pharmacogenomics. This is the future. Your liver uses a family of enzymes called cytochrome P450 to break down drugs. But everyone's P450 enzymes are a little different genetically.
So if my version of the enzyme is really fast, I burn through the warfarin too quickly.
Exactly. It might not have an effect, and you could still clot. If you have a slow version, the drug builds up to toxic levels, and you risk bleeding out.
Which is why doctors are now testing for these enzymes to personalize the dose.
It's personalized biochemistry, treating the patient's enzymes, not just their diagnosis.
Okay, let's talk about the engine that powers all of this, metabolism.
Right. Metabolism is a word we hear all the time. But scientifically, it's the sum of all chemical reactions. It really comes in two main flavors, catabolism and anabolism.
Break that down for us.
Catabolism is breaking stuff down. Think catastrophe. You break down food to release energy. Anabolism is building stuff up. Think anabolic steroids, building muscle. That requires energy.
And the king of the whole system is glucose.
Glucose is central. It's our primary fuel. We store it as glycogen.
But when the system breaks down, we get metabolic syndrome.
Yes, which is a whole cluster of issues.
Yeah.
It's not just weight. It's abdominal obesity, plus insulin resistance, hypertension. The works.
And the text highlights a specific hormone here, leptin.
Right. So leptin is the satiety hormone. It's made by your fat cells, and it's supposed to tell your brain, hey, we've got enough energy stored, you can stop eating now.
So in theory, someone with more fat should have more leptin and less appetite. But that's not what happens.
It's not, because of resistance. In obesity, the brain often stops listening to the leptin signal. So even though leptin levels are high, the brain thinks the body is starving.
And that creates what the book calls a high set point.
Exactly. The body fights tooth and nail to maintain that higher weight. It's why regaining lost weight is so common. It's not a failure of willpower. It's a broken biochemical signal.
Which, of course, leads directly to type 2 diabetes and heart disease. It's all connected.
Everything comes back to the chemistry.
So let's go to the final piece, the blueprint itself, genetic information.
The central dogma. DNA makes, RNA makes protein, the flow of information. But the really elegant part is how we figured out how DNA copies itself.
The Maselson-Stahl experiment, a classic.
It's maybe the most beautiful experiment in biology. They wanted to know, when DNA replicates, do you get one old double helix and one brand new one? Or does it split apart?
And they used heavy nitrogen to figure it out.
Isotope N15, yeah. They grew bacteria in heavy nitrogen, so their DNA was literally heavier. Then they switched them to light nitrogen and let them divide. And what did they find? After one generation, all the DNA was a hybrid density. Not heavy, not light, but right in the middle. It proved that each new DNA molecule consists of one old parental strand and one brand new light strand.
So it's semi-conservative replication.
We keep half of the past in every new cell. It's quite poetic, really.
And now, of course, we're not just observing DNA, we're editing it with CRISPR.
CRISPR-Cas9, yeah. The work of Doudna and Charpentier. It's a molecular scalpel that lets us make precise cuts in the genome.
And the application the book mentioned, it's just incredible. Malaria.
It is. Instead of just trying to cure the person with malaria, researchers are using this technology to edit the genome of the Anopheles mosquito itself.
To make the mosquito unable to transmit the parasite in the first place.
Exactly. If you can edit the mosquito's genes so it's resistant to the malaria parasite, you break the entire chain of transmission. You prevent the disease before it can even start.
Wow. From the 6 basic elements all the way to editing a mosquito's DNA.
It's the whole spectrum of life.
So to wrap up, we've gone from the big 4 molecules to hemoglobin, to how bones remodel in blood clots, to the engine of metabolism, and finally to the genetic code.
The real takeaway is that this isn't abstract science. It's the reason warfarin works. It's why braces straighten teeth. It's why smoking is so damaging. It's the practical science of health.
Okay, so before we let you go, as promised, a little review question for you listeners.
A pop quiz. See if you were paying attention.
Here it is. You have a patient who needs the blood thinner warfarin. Based on our discussion, why would that patient's genetic makeup, specifically their cytochrome P450 enzymes, influence the dosage you give them?
Take a second. Think about the liver. Think about breaking down drugs.
Got it. Okay, what's the answer?
The answer is all about the rate of metabolism.
Yep.
Those cytochrome P450 enzymes are what clear the drug from the body. If your genes make those enzymes work extra fast, you'll clear the drug too quickly and it won't work.
And if they're too slow?
If they're slow, the drug builds up in your system and you're at high risk for dangerous bleeding. So you have to tailor the dose to match the patient's personal biochemistry.
Perfect. It's always about the enzymes.
It is always about the enzymes.
Thank you for joining us on this deep dive into essential biochemistry. Hopefully you'll see the machinery of life a little more clearly now.
Keep asking how. The molecular logic is always there, waiting to be found.
See you next time.